I'm Jeremy Nathans. I'm a professor at the Johns Hopkins Medical School and an investigator of the Howard Hughes Medical Institute. This is the second of 3 lectures on the vertebrate retina, its structure, function, and evolution. In this lecture, we're going to look at human color vision and its variations. Now, why should we study color vision? Well, one reason, the reason I like most, is because it's beautiful. We see a world of color out there, as seen in this upper photograph, and if we were deprived of that color, the world would be a far duller place. And the story I want to tell you is really a centuries-long story. It begins in 1665 in the dormitory room of Isaac Newton, who was an undergraduate at Cambridge University at that time. And Newton did a very simple yet profound experiment. He drilled a hole in his wooden shutter to admit a beam of light, and he passed that beam of light through a prism and asked what happened to it. And the answer was the beam was split up into its component colors, the colors of the rainbow, from red at the least refrangible to violet at the most refrangible, and in between, orange, yellow, green, and blue. Now, that had been known for some time, but Newton appreciated the physical significance of that phenomenon. That is, that white light was a summation of different lights, and that those lights could be separated physically in an objective fashion, and that their properties could be objectified in the sense that each kind of ray had a particular angle of refraction when passed from air to glass and then glass back to air again. And furthermore, Newton realized that this method could be used to purify the individual rays of light to obtain spectrally pure samples with which to work. So, for example, by blocking off all but the orange ray of light, as shown here, Newton was able to obtain a beam of spectrally pure orange. Nowadays, perhaps we would do this with a tunable laser to obtain a spectrally pure light. But Newton appreciated the power of this purification method, and he used it to good effect. And in particular, he used it to recombine lights of different colors. Now, I can't resist showing you Newton's original sketch. This is his quick pen sketch of the experiment. It's reversed left to right from the image I showed you earlier, but here's the beam of light coming in the window. It's refracted by the prism, and then various beams are obtained from it by using their specific angles of refraction. And in particular, Newton performed the following very simple but profound experiment, diagrammed here essentially with projectors, as we might do it today, but Newton did it with mirrors and those beams of purified, spectrally purified lights. Newton asked, what if you combine together a spectrally pure red and a spectrally pure green light? What would it look like? The answer is it looks like neither. It looks yellow. That is, the superposition of red and green looks just like a spectrally pure yellow. But it is not physically identical to a spectrally pure yellow. And Newton could prove this by passing this yellow, such as we see here, through another prism, and a spectrally pure yellow, identical to it in all appearances, also through a prism. And what he saw was that they behave quite differently. A spectrally pure yellow retains its same initial angle of refraction and remains a single homogeneous beam when passed through a prism. But a synthetic yellow, such as we see here, one obtained by superposition of red and green lights, in fact splits up back into its component, red and green lights, each with their own original angles of refraction when passed through a prism. And Newton realized that this meant that human color vision was imperfect in the sense that it could not distinguish certain physically distinct combinations of lights. Now, as far as we know, Newton never investigated the physiologic basis for that phenomenon, although he worked on the physical rules that govern the recombining of lights of different colors. Now we would refer to them as different wavelengths instead of the angles of refraction that he used. But he had remarkable physical insights into the color vision system. And let me just give you one of these. This appeared in his Optics, his wonderful book describing his early experiments. And here Newton asks, may not the harmony and discord of colors, that is, the similarities and differences of the colors, arise from the proportions of the vibrations propagated through the fibers of the optic nerves into the brain, as the harmony and discord of sounds arise from the proportions of the vibrations of the air? Now, this is a remarkable insight at a time when it was not at all clear what the physical nature of light was. Newton had a sense, really by analogy, that light might consist of some sort of wave. And I should say that Newton also, in the optics, discussed the evidence that light has a particle-like characteristic. And in fact, he comes down somewhat in the middle and says that it is both particle-like and wave-like, a remarkable judgment in light of the fact that this is 2+ centuries before the quantum theory emerged to tell us that in fact light does have both particle and wave properties. Now, the physiologic underpinnings of the color mixing of the kind that Newton saw was not really laid out until the 19th century. Its first clear description we owe to Thomas Young, the universal genius who worked on many things, including the Rosetta Stone and vision. And Young, in the Bakerian lecture of 1802 before the Royal Society of London, suggested the following as a model for how color vision might work. And he says as follows: as it is almost impossible to conceive each sensitive point of the retina to contain an infinite number of particles, that is, an infinite variety of receptors, each capable of vibrating in perfect unison with every possible undulation, that is, every possible wavelength of light, because at this point it was clear that light had wave properties, it becomes necessary to suppose the number limited, for instance, to the 3 principal colors, red, yellow, and blue, and that each particle is capable of being put in motion more or less forcibly by undulations differing less or more from perfect unison. Now, this very prescient statement, in fact, hit the nail exactly on the head, because what Young is saying here is that each of the 3 receptors would have a broad bell-shaped curve. There would be a place where it is maximally sensitive, where it is matching the so-called undulation of the stimulus, and then there are other parts of the spectrum where it would be less sensitive because the match was less perfect. And so by having that sort of broad sensitivity, the limited number of receptors, the 3 receptors that he's postulating, would be able to cover the spectrum, and between them, and by comparing their degrees of excitation, one could determine the color appearance of any given light. That was exactly right. Now, the physiologic evidence, the behavioral evidence really from testing humans that would support this model was not forthcoming until another 50 years or so, and it came from 2 equally remarkable scientists, Hermann von Hemholtz, shown at the left, and James Clerk Maxwell, shown at the right. They're both shown here in their 20s. Helmholtz, at the time of this photograph, had just invented the ophthalmoscope and revolutionized ophthalmology. Maxwell, shown to the right, is holding his famous color top. He's an undergraduate in this photograph. He had been interested in color for many years, starting as a young man, and had devised a series of tops with different colored patterns that he could spin at high speed to effect a uniform mixing of the colors. We now know, if we can fast forward another 150 years or so, that the Young model is exactly right, and that there are in fact 3 light sensors, and they have broad bell-shaped curves, and these curves have now been identified by a number of methods, and here's just one of them. In this case, the individual proteins that constitute these receptors and their chromophores have been joined together, and the absorbance spectra determined in a spectrophotometer, and we see that rho, which stands for rhodopsin, the one in rods, is this curve peaking at about 500 nanometers. This one is not involved in color vision. I put it here just for completeness. It's involved in dim light vision only, but it has the same overall shape because these different pigments work in terms of their photochemistry really in the same way. But the other pigment curves, the longer wave, the so-called L curve, the medium wave, the M curve, and the short wave, the S curve, are offset by substantial numbers of nanometers from one another and from the rhodopsin curves. And together they span essentially the full range of the visible spectrum. Now, what does this get us in terms of color vision ability? Well, we can ask that in a number of ways. One way we could ask that question is by simply asking, at any given wavelength along the entire visible spectrum, how different do two lights have to be to tell them apart? So that's shown here, for example. Here on the horizontal axis is wavelength, and on the vertical axis is the differential, the delta lambda, the differential between two lights that one needs to discriminate them. I think you can appreciate that the curve is not a simple flat curve. It goes up and down. There are hills and valleys. There are some places, for example, right here, where a difference of perhaps half a nanometer is sufficient to distinguish two lights, and other places where a difference of, say, 6 nanometers or so is required. This curve of hills and valleys is completely understood by reference to the original absorbance curves of the pigments. So, for example, if we ask how we can distinguish, say, a light that is at, say, 600 or so nanometers, we can distinguish it because it differentially excites the long and medium wave pigments, and a light of, say, 595 nanometers, a little bit blue shifted relative to it, has a different ratio of excitation of those two pigments. And that's a sufficiently great difference that we can tell them apart. I should add that this set of curves also, of course, explains Newton's color mixing data in a very simple way. For example, if we have a sum of red and green lights, those give a certain degree of excitation of those three different sensors that is the same, as it turns out, as a spectrally pure yellow light. So, of course, the system cannot tell them apart, because in the end, a detector is really just a photon counter. Once a given number of photons is captured and that number is then sent to the brain, there's no way to tell what the source of those photons were. Was it a long wave light that was captured inefficiently but was very bright, for example, or was it a shorter wave light which was captured less efficiently but was dimmer? That's completely lost within the system. And any pair of lights which give the same total number of photons captured per unit time for each of the three receptors will, of course, be then indistinguishable. As part of Maxwell's experimental approach to analyzing human color vision, he brought to us a conceptualization of color perception, which is extremely useful and is shown here. Maxwell thought that we could conceptualize color space as a three-dimensional space in which each of the three axes, as shown here, the L, the M, and the S axes, correspond to the degrees of excitation of each of the three classes of cones. So, for example, a white light, which equally excites all three cone classes, as shown by this corner of this square here and this vector coming from the origin, would be represented by a vector in this direction, and lights of other wavelengths would be represented by distinctive vectors in their own unique directions. So in Maxwell's conceptualization, our color vision is a three-dimensional color vision. We have 3 degrees of freedom. If an individual were to have only 2 receptors, that individual would have a 2-dimensional color space. So for example, if one is missing the L receptor, the color space would consist of the M and S axes and the plane defined by them, but not the 3-dimensional S, M, and L space. Now let's talk a little bit about the chemistry of color reception, and in particular the ways in which different wavelengths of light might be selectively absorbed by the different visual pigments. We saw in the first lecture the overall structure of a visual pigment. It's a G-protein coupled receptor. It has 7 transmembrane segments and sits in the membrane of the outer segments. And as you recall from that first lecture, the actual light absorbing part is a small molecular weight vitamin A derivative called 11 cis retinal. That's true for all of the human pigments. They're all built on the same basic architecture, and they all use exactly the same 11 cis retinal chromophore. So the difference between the pigments is a difference in the proteins, and what the proteins do is they impose a somewhat different electronic environment around that retinal chromophore, which has the effect of tuning its absorbance spectrum to either longer or shorter wavelengths. So one can think about this spectral tuning challenge as really a question of how to modify the pi-electron system of the retinal chromophore by virtue of the surrounding amino acid side chains. And we can learn something that at least constrains the possible mechanisms by examining the basic photochemistry of retinal itself. The critical point with respect to spectral tuning is that the degree of pi-electron delocalization within retinal determines its absorbance spectrum. Now, retinal, being a conjugated polyene, as shown here with 6 double bonds, has a substantial amount of resonance and therefore delocalization of electrons. I've shown below just one resonance structure that one could draw, not a particularly favorable one because it places a positive charge on this carbon here as a result of moving the pi electrons over towards the nitrogen, which now has a free electron pair. But this, of course, sort of resonance structure, even though it is relatively unfavorable, will contribute to some extent in that equilibrium ensemble of all resonance structures. Just to get a sense of how substantial this resonance effect is in the absorbance spectra of these kinds of compounds, let's look at 2 model compounds, which are shown here, a polyene at the top and a cyanine at the bottom. Now, these are drawn with the same number of double bonds as retinal has, that is 6. And I think you can see from, if we look at this polyene at the top where the chain ends in carbons, here that a resonance structure of the sort that I've drawn just below it, in which one carbon now carries a positive charge and the other carries a negative charge, would be quite unfavorable. That involves, of course, a charge separation in addition to the general unfavorability of placing charges on carbons. And so, in general, polyenes have very little pi-electron delocalization. We know this for a fact because when one looks at the crystal structure of a polyene, one sees that the lengths of the bonds are highly alternating. There are lengths that are very long, the canonical single bond lengths, and there are others in between that are very short, the double bond lengths. And so this back and forth, long, short, long, short, long, short, tells us that really the single bonds have virtually exclusive single bond character, the double bonds exclusive double bond character. And to go along with that, the absorbance spectra of the polyenes is at quite short wavelengths. For example, here we're down in the ultraviolet at about 360 nanometers for the absorbance maximum. At the other end of the spectrum, so to speak, for pi-electron delocalization would be a model compound of the sort shown down here. This is a cyanine, again, 6 double bonds, but now we have an NH2 at both ends. In one case, there's a positive charge, in the other case, a free electron pair. And I think you can appreciate that the sort of resonance structure that is drawn below, in which each single bond has now been switched to a double bond, each double bond now switched to a single bond, simply is a mirror image of the one up above. Now the free electron pair is at the right, the positive charge is at the left, but everything else is the same in chemical terms, and therefore these two must be equal in their energies and therefore equally likely to occur, and therefore the pi electrons are maximally delocalized in a cyanine type compound. And again, we know this is true because from crystal structures of cyanines, we see that all of the bond lengths are the same, and they're midway between the lengths of single and double bonds. And going along with that, we see that the absorbance maximum of a cyanine is quite long. In this case, it's 750 nanometers, far to the red. This would be a very deep red compound. So with these 2 model compounds, we see a difference in absorbance that spans essentially the entire visible range. All visual pigments that are known in nature are somewhere within the extremes defined by these two model compounds. And in essence, what visual pigments are doing is they're creating, with the retinal chromophore, some sort of hybrid between a polyene and a cyanine, a hybrid in the sense that the electrons are delocalized to a degree that is intermediate between the delocalization seen with a polyene and the delocalization seen with a cyanine. And correspondingly, the absorbance spectra is intermediate between the extremes shown here, 360 nanometers and 750 nanometers. So now that focuses our thinking a little bit, because really what we can imagine the protein must do is make the chromophore look more polyene-like or more cyanine-like. How it does that, we don't know in complete detail at this point, but we have a few clues. First, we have the amino acid sequences of the proteins. And when we look at those sequences, and here they're shown with the proteins splayed out in 2 dimensions, with each dot in each of these schematics indicating an amino acid difference. So here, for example, we're comparing the M-cone pigment, the middle wave sensitive pigment, with rhodopsin. Here we're comparing the S, or short wave pigment, with rhodopsin. Here we're comparing the medium pigment, the M pigment, with the short wave pigment. And here, and the lower right, we're comparing the medium pigment with the longer wave pigment, M versus L. So I think you can appreciate that although they have the same overall structure, there are 7 transmembrane G protein coupled receptors. There are obviously many amino acids that are identical. The open circles indicate identities. There are also, for at least 3 of these comparisons, 3 of these pairwise comparisons, many amino acid differences. But I think you can also appreciate there's an asymmetry here. The M versus L comparison shows that these 2 pigments are extremely similar one to another. There are only about 15 amino acid differences, depending which variants. There's a few allelic variations that can make that number a little bigger or smaller, but roughly 15 amino acid variations, amino acid substitutions that distinguish the M and L pigments out of a total of 364 amino acids. That asymmetry is going to be the focus of much of the remainder of this lecture and the next lecture. Now let's just look at what that sequence difference looks like in a dendrogram form. Here we're looking at sequence divergence among human and mouse rod and cone pigments. We've picked the mouse as a representative non-primate mammal. And here let me just say that the non-primate mammals have only 2 types of cones and rods, very much like our rods, whereas a subset of primates, like ourselves, have 3 cone pigments. And now if we look at these amino acid sequences, and here we're plotting on the horizontal axis the degree of sequence divergence, the typical dendrogram type plot, and on the vertical axis we're plotting the wavelength of maximal absorbance of the corresponding pigment. So let's just walk through this, starting with the mouse example. The mouse rhodopsin, like typical mammalian rhodopsins, absorbs at about 500 nanometers. And its sequence is rather different. That is, this dendrogram line is rather long, this horizontal line, compared to the mouse medium wave pigment, the only example that the mouse has of a longer wave sensitive pigment. And it's also quite different from the shorter wave pigment that the mouse has, which actually absorbs in the near ultraviolet. It's absorbing at wavelengths somewhat shorter than the one that we have. In the case of the human pigment set, we see that, again, we have a rhodopsin typical of mammalian rhodopsins, and we have a shorter wave pigment, and these differ substantially in sequence. And then in what must have been a very recent evolutionary event, there has been a split on the arm of the tree to give a long and a medium wave pigment, whereas our immediate mammalian ancestors did not enjoy that pair of pigments. When the genes for the long and medium pigments were first identified, not only were their sequences found to be very similar, and that of course is the source of the sequence comparison that we saw just a few slides ago, but also the arrangement of genes was such that they were found to be adjacent to one another on the DNA. So here is the human arrangement of a long wave pigment gene shown with a red arrow and a medium wave and M pigment gene shown with the green arrow. And that's not a surprising arrangement given their recent evolutionary history. Genes that duplicate are not infrequently found to be adjacent to one another. And we imagine that at some point in evolution there was a erroneous recombination event which paired a segment downstream of one gene and upstream of another. And as a result, we got a chromosome, or our ancestors got a chromosome with both genes adjacent on the DNA. Now I've drawn here one aspect of that ancient arrangement, which I'll justify in the third lecture, and that is that the original pair of pigments which underwent this recombination were actually not identical. One was already an L pigment gene and the other was already an M pigment gene. That is, they were already different from each other at the time of the duplication. And let me reserve that, as I said, that story for the third lecture. Now, if we look in modern day humans, although we see this arrangement of a single L and a single M pigment gene on some chromosomes, in fact, only about 20% of human X chromosomes, it turns out these are on the X chromosome, only about 20% of human X chromosomes actually have that arrangement. When DNA from a large number of different humans was examined to look at the arrangement of the L and M genes, it became apparent that the basic arrangement of one L gene and one M gene, as shown at the top here, is actually not the most common of arrangements in the population. In fact, most people have one L and two M genes. Some have one L and three, and there are a few even who have 4 or 5 M pigment genes. All of these people, as far as we can tell, have perfectly normal color vision. Now, this arrangement almost certainly arises because the genes are homologous to one another and are capable of recombination, and we'll discuss that in just a minute. This recombination, in fact, gives rise to a number of variations in gene structure, and the sort of variation which appeared first in terms of scientific attention was the kind that affected color vision and has been called, perhaps incorrectly, color blindness. Now, color blindness doesn't mean a complete inability to see colors as generally used. It means simply a reduction of color vision from trichromacy, from having 3 receptors down to having only 2. Perhaps the most famous trichromat is shown here, John Dalton, the great British chemist, who in his first scientific paper in 1794 described his own color vision in meticulous detail. Dalton was a dichromat, and he reported that what other people called red or orange or yellow or green appeared to him to be just a single kind of color, differing only in intensity. Let's see what the world looks like to people who are missing one of the 3 receptors. And I'm showing here photographs of a fruit stand taken and processed so that we see what it would look like to a normal trichromat here on the upper left, or to any of the 3 types of dichromats. On the upper right is so-called protonopes view. That's someone missing the long wave sensor. On the lower right is the deuteranopes view. That's someone missing the medium wave sensor. And over here on the lower left is a tritonopes view. That's someone missing the shortwave sensor. Now, I think you can appreciate that, as Dalton reported, without the long or medium wave sensor, the fruit looks very much washed out in its colors. That is, the longer wavelength colors, the ones that particularly characterize the ripeness of fruits, the green, yellow, orange, and red colors, really are not distinguishable readily by an individual who's missing either the long or the medium wave pigment. What's the origin of these not uncommon color vision anomalies? It turns out that they arise from unequal recombination between the members of the L and M tandem array, and that this recombination occurs quite readily because the genes not only are adjacent to one another, not only are they in a head-to-tail tandem array, but they also have retained a very high degree of sequence identity, approximately 98% identity at the DNA level. And so as a result, mispairing can occur at meiosis of the kind shown here. For example, a region distal to this M pigment gene is nearly identical in sequence to a region just distal to this L pigment gene. And if those two regions were to pair at meiosis and a recombination event were to occur there, we would get the two meiotic products shown at the bottom, one in which only a single L pigment gene remains, and the other in which an additional M pigment gene has been acquired. The one at the top, if inherited by a man, since this is on the X chromosome, for whom this is, of course, the only X chromosome, would cause a dichromacy. This gentleman would be missing M pigment and would have only the S and L pigments. As far as we know, as I mentioned earlier, people with extra pigment genes, extra M pigment genes, have perfectly normal trichromatic color vision. In fact, current evidence would suggest that only the first gene in the array, when there are multiple M pigment genes, only the first M pigment gene is expressed, and more distal M pigment genes are not expressed. Now, what if recombination occurs at an intragenic location rather than between the genes, that is, within the genes? Now, well, if this region of the M pigment gene pairs with a corresponding region of the L pigment gene, and again, these regions are almost identical in sequence, we would create meiotic products in which there would be hybrid genes. So, for example, one product in which the M pigment gene was lost would retain not a normal L pigment gene, but an LM hybrid, and the reciprocal product would be present on the other chromosomal product of this recombination with an M pigment gene picked up as a more distal gene in that recombination event. Now again, the color vision of these individuals would be affected. Of course, the person who inherits this chromosome at the top, the male who inherits that chromosome, would be missing one of the longer wave genes, and in this case would have the LM hybrid instead of either a normal L or a normal M gene. In the case of the individual who gets the chromosome shown at the bottom, recall that I just mentioned that only the first two genes in the array are expressed, so this person would express the L pigment gene, that would be a normal gene, but the ML hybrid would now be expressed instead of a normal M pigment gene. And that hybrid, we've come to realize, would in general have an absorbance spectrum different from either of the two parental pigments. So this hybrid, and this is also true of the hybrid diagrammed here, would be shifted in its absorbance spectrum, and the individual down here would be an anomalous trichromat, one who had three pigments, but not the normal set of three. And this person would make different judgments when asked to quantitatively mix different colored lights. Now, more complex events are also possible because many of the starting arrangements involve arrays that have multiple M pigment genes. So, for example, if we start with an array that has two M pigment genes and a second array that has only one, and envision the kind of recombination events, in this case intragenic recombination events, that one might encounter, we can, for example, see a meiotic product that has a hybrid gene, an LM hybrid instead of the L pigment gene, paired with an M pigment gene. Now this person would also be an anomalous trichromat, in this case having replaced the normal L pigment gene with a spectrally shifted hybrid. And the person down below would be the reciprocal kind of anomalous trichromat, now having a hybrid ML and an unexpressed M paired with a normal L pigment gene. These are quite common in the population. Roughly 8 or 10% of human X chromosomes have some sort of rearrangement that affects trichromatic color vision, and therefore roughly 8 or 10% of males who carry those chromosomes show that color vision anomaly. There are also more subtle variations in color vision, and let me show you the one that is the most common and perhaps the most fascinating, and that is a single nucleotide variation which divides the apparently color normal population into two distinct perceptual groups. This is among males, and among females into three perceptual groups. This is a single nucleotide difference in the L pigment gene, which results in a single amino acid substitution. It's a serine versus alanine difference at position 180. And if you look at, in this psychophysical test a mixing of red and green lights, and here the result is plotted on the horizontal axis as a ratio of red to red plus green lights, and on the vertical axis the number of individuals who had a given test result, and here the mixture of red and green lights is being compared to a standard spectrally pure yellow light, in just the way that Newton did this color mixing experiment. What we see is that some people, let's look at the male curve at the top, males are at the top, females at the bottom. Some males require more red in the mixture to match the standard yellow. Others require less, and the distribution is bimodal. And in the female set, there are some that test like the males who require more red, some who test like the males who require less red, and others who are in the middle. These are the heterozygotes in the middle. And what we're seeing is the effect of this polymorphic variation and its effect on the absorbance spectrum of the red of the L pigment, which is now shown here, determined analytically by producing the recombinant proteins and reconstituting them with the retinal chromophore. So the serine version, the one that has serine at position 180, is a little bit redshifted, that's this upper curve, compared to the absorbance spectrum of the alanine at position 180. This is a 4 or 5 nanometer shift, and it precisely accounts for this variation in spectral sensitivity between individuals who have either the alanine version or the serine version of the L pigment. So even though we think, perhaps simplistically, of the world of trichromats as a single unitary group of individuals with respect to their color vision, that's not so. Individuals are seeing the world somewhat differently based on this genotypic difference. And although each of them is internally consistent, if you gather them together in a laboratory and ask them to quantify color mixing, you'll see that they divide up into these distinct perceptual groups. We saw at the beginning of this lecture, and also in the last slide, how human visual pigments can be produced in the laboratory and their absorbance spectra measured directly after they are joined to 11-cis retinal. That's a wonderfully direct technique, and it gives us a very precise measure of the absorbance spectra of those pigments. Now we'd like to ask, if we go back to the living human eye, can we analyze the pigment spectral sensitivities in the same precise way? And in this example, I will show how males who have only a single gene on the X chromosome provide a perfect group for this sort of study. So these gentlemen have lost by homologous recombination all but one of the genes on the X chromosome. What remains behind is either a normal L pigment gene, because they've lost the M pigment genes by homologous intergenic recombination, or what remains behind is a single hybrid gene containing either more or less of the parental contributions depending on the exact point of recombination. But again, the other genes in the array have been lost by this recombination mechanism. And these subjects are, in a sense, the perfect ones to study because they have only 2 cone pigments that one needs to examine, the S pigment and the remaining longer wave pigment. And for simplification, one can design the study such that the S pigment is really not in any way interfering with the sensitivity measurements for the longer wave pigment. So one can use longer wave lights and selectively desensitize the shorter pigments. So in a sense, we have in at least the longer wave end of the spectrum, we have the very simplest arrangement one could possibly have. We have someone who has just a single X chromosome, and on that X chromosome just a single visual pigment gene. And we're going to ask, what is its spectral sensitivity? And that is shown on this next slide. This is determined by a method called flicker photometry. It's a completely safe and non-invasive method. The individual views a screen on which is alternating about 20 times per second a standard light, a light of a standard wavelength, say 600 nanometers. And with it, in alternation, is a test light whose wavelength is variable. It could be 610 nanometers, 620, 630, 640, 650, and so on, in 10 nanometer increments up and down the wavelength axis. And what the subject has to do is to adjust a dial which controls the intensities, the relative intensities of these 2 lights, so that the sense of flicker is completely eliminated. Now let's ask how that could possibly happen. After all, these are two differently colored lights, lights of different wavelengths that are being presented, alternating one with another. How could the sense that they're flickering back and forth not be seen? Well, remember, these subjects have only a single visual pigment in the long wavelength end of the spectrum. And we've suppressed the S-cone pigment so that it's really irrelevant to this measure. And I should say the rods have also been suppressed by the high intensity of the lights being used here. So for that one remaining pigment, if the number of photons captured per second by these two different stimuli is the same, then at that point, for the individual, there's no sense of flicker. It just looks like a continuous illuminant. And that's the sense that is obtained when the dial is set to just the right location. I think you can appreciate that that gives us the relative efficiency of photon capture for those two different stimuli. And that, of course, is done for each of the different test wavelengths along the whole wavelength axis, and then we can plot out the full sensitivity curve. And here they are. In green, we see the curve of sensitivity for the M pigment. That is, these are individuals who have an LM hybrid, but the LM hybrid consists almost completely of M sequences. And in particular, it consists of M sequences in those regions which are critical for the spectral difference between L and N pigments. So de facto, these are individuals who have only an M pigment and no L pigment. And here is their sensitivity spectrum in green. And then in yellow and red are the two different variants of the L pigment, one that has alanine at position 180 or serine at position 180. And I think you can appreciate that the serine at position 180, just as it was in the in vitro spectrum, is offset to the right compared to the alanine at 180 version. So here, this is a very satisfying result. In the living human eye, we can see the full spectral sensitivity of the pigments that underlie normal color vision in the human population. And my only regret in describing this experiment is that that most interesting of dichromats, John Dalton, didn't live long enough to participate. Dalton was born in 1766. I think if he had been born 2 centuries later, he would have been an eager participant in this experiment. Dalton maintained a lifelong interest in color vision and color blindness. And even though he didn't participate in this experiment, you may be interested to know that he made substantial contributions to the science of color vision, and in particular to understanding color blindness, even after his death, which seems surprising. But Dalton suggested during his lifetime that perhaps the source of his color anomaly was a selective filtering out of longer wavelengths of light by some colored medium, either in his vitreous, in the fluid within the eyeball, or perhaps in the lens. And so he stipulated in his will that when he died, his eyes should be cut out and a little hole cut in the back of the eyeball. And by viewing the world through that hole, it would be possible to tell whether the world was being filtered by some sort of color filter within his eyes. And so that was duly undertaken upon his death. And the result was that except for a little bit of yellowing of the lens, which is to be expected in someone who's somewhat older, the path was completely clear. There was no filtering at all by the hypothesized color filter. And therefore, Dalton's color blindness must have been due to some difference within the retina or possibly the brain. And this was a major conceptual leap in understanding the origins of color blindness. And it would have been a substantial contribution if nothing more had been done. But in fact, Dalton continued to make contributions to the world of color blindness research. And 150 years later, his eyes, which had been preserved by the Manchester Literary and Philosophical Society, were examined by DNA analysis. And a small tissue sample was taken for that analysis. And it was shown that Dalton was in fact missing the M pigment gene. And he was a dichromat of just the sort we have seen before, who has only an L pigment gene and an S pigment gene. I think Dalton would have been very pleased with that result.